Hello everyone, welcome to the ITLS Academy. Today the topic that we are going to discuss is detection of Shigella. Okay, so again, why it is important to detect the Shigella into a sample? We all know that if the contamination of Shigella occur into the sample, then it will affect the health of the human being, because the Shigella is what? This is a microorganism and it will spoil the product. That's why it is very important to detect this Shigella into the sample. So in today's lecture, we are going to see that how we detect this Shigella, which medium we are going to use, because when we are detecting the microorganism into a sample, then our focus is on the media. Media means that the substance at which the microorganism can grow. Okay, so in case of Shigella, we are using a different media so that it will support the growth of that microorganism, that is your Shigella. uh, so let's start today's lecture. The first thing is your introduction. We all know that Shigella is what? This is a gram negative. We all know that bacteria are of two types. One is your gram positive and one is your gram negative. Okay. And how we find it that the particular bacteria is gram negative or gram positive? We perform a gram staining. Okay. So if purple colonies we get, then the bacteria is what? Gram positive. And if we get a pink colonies, then it means that the bacteria is what? Gram negative. Okay, so the Shigella show a pink color colonies when we perform a gram staining under a microscope. That's why it's called a gram negative bacteria. Aerobic, non-spore forming, non-motile, rod shaped bacteria. Okay, then principle detection of Shigella is totally based on pre-enrichment, selective enrichment, which is followed by the isolation on a selective media. Presumptive Shigella colonies are identified by a biochemical and serological test, and this pre-enrichment broth enable injured cell to grow. A selective enrichment is very necessary so that it will increase the proportion of the Shigella species in relation to background flora. This is the principle at which this experiment are performed. Now the next is your culture media. Culture media means that the, in simple word, it is the food of the microorganism, or you can say that the substance at which the microorganism can grow. So here we use different media, that is your nutrient broth, coffermin molar tetrathionide broth, selenite F broth, deoxy chocolate citrate agar, McConkey agar, and nutrient agar number 22. Okay, so first of all, just see the composition of that nutrient agar. Composition of nutrient agar. Okay, so we use a beef extract, then peptone, okay, then sodium chloride and distilled water. Okay, if the distilled water is 1000 ml or 1 liter, then the beef extract will be 3 gram, peptone will be 5 gram, and sodium chloride will be a 5 gram. This is the composition for the nutrient agar. Now the next composition is for the coffermin molar tetrathionide broth. Okay, so it contains peptic digest of animal, peptic digest of animal, and casein enzyme, enzymic hydrolysate as a source of carbon, vitamin, nitrogen, and minerals. Vitamins, nitrogen, and minerals. Okay. Next is your selenite F broth. So the composition of this selenite um, F broth is what it contain: enzymatic digest of casein, enzymatic digest of casein, and it also contain lactose, sodium phosphate, sodium phosphate, sodium hydrogen selenate, sodium hydrogen selenate, and that's it. Okay. Now this composition is again for a 1 liter. So enzymatic digest casein will be 5 gram per liter. Lactose will be 10 gram per liter. Sodium phosphate will be 4 gram per, sorry, lactose will be 4 gram per liter. 4 gram per liter. Okay. Sodium phosphate will be 10 gram per liter and sodium hydrogen selenate will be 4 gram per liter, and the final pH will be 7.0 plus minus 2. Okay. After mixing all these ingredients into a distilled water, what we have to do? We maintain the pH of the product, and the pH of the product should be 7.0 plus minus 2. Next is your decoxy chocolate citrate agar. Okay, so it contain different ingredient, and the ingredients of the this one, that is your now the composition of this deoxy chocolate citrate agar is what it contain: sodium citrate. It also contain protease peptone, lactose, sodium deoxy, sodium deoxycholate. Okay, so sodium, the amount of the sodium citrate will be 20 gram per liter. Proteose peptone will be 10 gram per liter. Lactose will also be 10 gram per liter, and sodium deoxycholate is around 5 gram per liter. It also contain a ferric ammonium citrate, okay, and neutral red, and finally it contain agar. Amount of agar is around 13.5 gram per liter. Neutral red is around 0.02 gram per liter, and ferric ammonium citrate will be 2 gram per liter, and the uh, final pH of that will be around 7. Okay, this is about the deoxycholate citrate agar. Now the next is your McConkey agar. McConkey agar contain a different ingredients, and the list of that ingredients are as follow. McConkey Okay, it contains peptone, protease agar, sorry, protease peptone, protease peptone, bile salt, crystal violet, bile salt, crystal violet, agar, lactose, sodium chloride, lactose, sodium chloride, and neutral red. Okay, so peptone will be 17 gram per liter, protease peptone will be 3 gram per liter, bile salt will be 5 gram per liter, crystal violet will be 0.001 gram per liter, agar will be 13.5 gram per liter, lactose will be 10 gram per liter, sodium chloride will be 5 gram per liter, and neutral red is around 0.030 gram per liter. And the last ingredient, the last culture media is what? It is a nutrient agar media, and the composition of this nutrient agar media is same as we discussed in a earlier lecture. So this is about the culture media. Then after preparing the culture media, we maintain the pH of all the media. Then we what we do, we do a autoclave. Autoclave should be done at 121 degrees Celsius, 15 psi for 20 minutes, so that all the microorganism present into it will be eliminated. Okay, so this is about the culture media. Next is your procedure. Okay, so the first thing that we do, pre-enrichment. In pre-enrichment, what we do, we clean the bottle, surfaces of the bottle, cup containing in pre-enrichment, what we do, we clean the surfaces of the bottle, pouches, cup, in which the sample are present with the help of the 70% of the ethanol. Then after that, we filter it with the help of the filter paper, whose pore size will be around 0.45 micrometer. And then after that, we assemble and then place this filter paper into a 50 ml of nutrient broth, and after placing it into a nutrient broth, we incubate it at 37 degrees Celsius for 18 to 24 hours. Okay, then after that, the second procedure is your selective enrichment. Okay, in this process, what we have to do, we transfer 1 ml from the pre-enriched nutrient broth into a 100 ml of selenite F broth and tetrathionate broth. And what we do, we take a 50 ml of selenite F broth and 50 ml of tetrathionate broth, and then after combining it, it will become a 100 ml. Then we incubate it at 37 degrees Celsius for 24 hours. Then after incubation, streak out a loop fold from a selenite F broth and tetrathionate broth, deoxycholate citrate agar. Now what we do, we make a streak, and when we preparing a streak, then in that case we take a media, that is your deoxy chocolate citrate agar, and the streak will be made from a selenite F broth tetrathionate. Then we incubate that plate at 37 degrees Celsius for 24 hours. If there is no typical suspendal growth will take place on DCA member, then again we re-incubate further for our 24 hours. Now the next is your observation. If there is a Shigella colonies present on deoxycholate citrate agar, then it will appear like a opaque with a ground glass appearance with even a margin. Okay, so confirmation test will be performed. If there is any typical suspended colonies are there, then what we do, we take a pick up the 5 colonies from each plate, and then we streak it into a McConkey agar. Suppose that this is your plate. Now in this plate there is a suspected colonies. Now what we do, we take up 5 colonies from them, from each plate, and then we make a streak into a McConkey agar, plate. Okay, now we take a plate in which we already put the McConkey agar. Then we incubate this plate at overnight at 37 degrees Celsius so that we isolate the pure colonies. Then these pure colonies are transferred into a nutrient agar number 2 prior to a biochemical and serological confirmation. Okay, now the next is your biochemical confirmation. So for biochemical confirmation, the first test that we perform is gram staining. So typical colonies of Shigella, we all know that they are gram negative, and how we perform a gram negative test, that we discussed in a earlier slide, that we perform a gram staining with the help of the crystal violet and all these things. And even there is a separate lecture of this gram staining are already uploaded in our account. You can go and check the video related to this topic. Now the next is your motility test. Nutrient agar tube are inoculated by stubbing with a straight wire to a depth of 5 mm and then incubate at 37 degrees Celsius for 18 to 24 hours so that we check the motility. If there is any outgrowth in whole tube, indicate that the motility, and if no motility is observed within a 24 hour, then what we do, we place the tube at an incubator at room temperature for further 4 to 6 days, so that motility will be detected, because Shigella is not motile. Now the next is your catalyst test. Catalyst test is performed. We take a streak colony on a nutrient agar slab and we incubate it at 37 degrees Celsius for 24 hours. And after that, we take a colonies and add 1 ml of hydrogen peroxide at that growth. Okay, Shigella is a catalyst positive, and if there is oxygen is released from the plate and bubbles are formed, then it means that hydrogen peroxide um, will be eliminated as a form of the bubble, and it will indicate the presence of the catalyst and the presence of the Shigella. Now the next text is your oxidase test. In that case, what we do, we streak a colony on a nutrient agar slab and we incubate it at 37 degrees Celsius for 24 hours to a nutrient agar slab containing the culture. Add few drop of the mixed text reagent, 1% of the alpha nephthol in 95% of ethanol, equal amount of the para amino dimethylene and aniline hydrochloride in a water. Then Shigella, being an oxidase negative, does not develop blue color with 2-minute addition of the reagent. Now, expression of the test, the presence and absence of the Shigella species given after a biochemical and serological confirmation of presumptive colonies in a sample. So, test for the Shigella, again, it is represented as a present and absence per x ml of sample. Suppose that there is 100 colonies in 250 ml, then we represent it like this: 100 CFU per 200 ml of the sample. So this is all about the determination of the Shigella. I hope you will understand, but if you have any query related to this topic, you can ask me in a comment box, or for a further detail you can follow the ITLS Academy on different platforms, that is Instagram, Facebook, Twitter, YouTube, WhatsApp and LinkedIn, and subscribe our channel ITLS Academy on YouTube and get our app from a Google Play, or you can also ask your query to the helpline number that are already mentioned in your slide, that is 7080833450, and visit our website www.itlsacademy.com. Thank you for watching. Please like, share and subscribe our channel.